Hello and good morning, biology. Happy Wednesday. Uh, sorry I'm out again today. Still not feeling well. I'm hoping to be back tomorrow. I heard you guys didn't quite finish the video yesterday, so make sure that you finish that video before you start this one. The assignment you can do all together at the end. Today we're going to start talking about Punnett Squares, and these are things you should have seen in middle school. If you didn't, don't worry. We're going to talk about them again, and we're going to get through them, and you guys are going to be experts by the time. You guys are going to be really good at Punnett Squares. Okay, let's get started. Today's video is shorter than yesterday's, promise. All right, so when we talk about Mendel's traits, and we talked about true breeding versus non-true breeding, something we need to talk about is genotype, which is an organism's genetic makeup. This is what your DNA says about you, or what an organism's DNA says about it. It consists of these alleles, or these factors, or these traits that an organism inherits from their parents. For purple flower, if you have the big R, that dominant, you would have a nice purple flower. Bloop. And if you have the little r, you would have a white flower. And I can't draw a white flower there because it wouldn't show up very well. But I drew white flowers all over this slide, so you just can't see them. They're just hidden. But so genotype is what the genes say. It's different than phenotype. So in phenotype, this is the physical expression of a trait. Think pH equals physical. So that dominant trait can hide recessive ones. In this example over here, I'm using P's in mine, they're using B's in theirs. Big B is homozygous dominant, meaning that it has two of those purple ones. It's a purple one. The heterozygote, the big B, little b, it is purple as well. Even though they're genotypes, what their genes are, they have different alleles, they are the same phenotype. And then if I have this homozygous recessive, little b, it is white. So even though these two, I'll circle them, bloop, have different genotypes, they have the same phenotype. Now homozygous versus heterozygous. In homozygous, it'll have both alleles of a pair are alike. So an example, the big P, excuse me, or the little p. But it's both dominant or both recessive. Like this one here, this is homozygous dominant, this one's homozygous recessive. Those are homozygous. Heterozygous is when the alleles in a pair are different. So this big P, little p, that's the heterozygote. Probability, this is the likelihood that a specific event will occur. This is the first time we've talked about probability in science in my classes. You guys will talk about it more if you take my microbiology. We'll talk about it more when we talk about Henry Weinberg equation coming up in a couple chapters. Sorry. The dominant trait happened 3 out of 4 times. So if we divide that, 3 out of 4 is 75% in his experiments. The recessive then was 1 out of 4, or the recessive, little r, would have been 25%, or 0.25, if you're talking about a probability. So we're going to do some monohybrid crosses today. You guys are going to be working on some. In monohybrid, only one characteristic is tracked. That mono means one. We're going to use these Punnett squares. I have a picture of one over here on the right-hand side for pod color. This is a diagram used to aid in the predicting of inheriting traits. Now, Punnett squares aren't guaranteed. If you have 4 kids, it does not mean you're going to get one from each box. It means that you have a 1/4, a 25% to have a kid from each box. So in example 1, we're going to do a homozygous times a homozygous. So we're going to use that nice pod color one, big G, and then little g. Let's see if this shows up. I'm going to use this more orangey color. I think it'll show up a little bit better. Little g. So in a Punnett square, you're going to grab mom or dad can go on the top as long as the other one goes on the side. You're going to bring this big G down into both of these spots. This one goes down. This one goes down in these two. And then these little g's come across. Let me find my... I'm going to use a different color next time. Little g, and then little g, little g. So, 100% of these, or 4 out of 4, have the genotype, big G, little g. We also have 4 out of 4 with the phenotype of green pods instead of yellow. So even though these guys are all heterozygotes, meaning they have one dominant and one recessive. So now let's do that same example. I'm going to use black instead for my pods. And we're going to do big G homozygous, big G, big G. Let's do, actually, to make this a little bit more exciting, let's do our heterozygote is big G, little g. And our homozygous is going to be homozygous recessive. So that means little g, little g. So same thing, big G's get pulled across, little g's go there, this little g gets pulled down, this little g gets pulled down. Now we start to see some interesting things. We start to see 2 out of 4, or 50%, are heterozygotes, or GG, and 2 out of 4 of them are homozygous recessive of GG. So what this means for phenotypes is we're going to have 2 that are green, we're going to have 0 that are homozygous, dominant, we'll have 2 that are heterozygous, and we'll have 2 that are heterozygous recessive. And so a way to remember this is when we do these phenotypes, we'll do homozygous, dominant, then you'll do the hetero one, and then you do the homozygous recessive. So 0, 2, 2 in this case. All right, now we have 2 heterozygotes. We have big G, little g, and big G, little g. And once again, we're going to drag these across. Big G, little g, little g. Pull this big G down, big G. Pull this little g down, little g, little g. Now, this one gets really interesting. We have this one that is homozygous dominant. And then we have 2 that are heterozygous. This one and this one are the same thing, even though their things are reversed. They look the same, they act the same, they are the same, even though their letters are reversed. In fact, you can rewrite this one as big G, little g, if that works for you better. And then we have one that is going to be the yellow, little g, little g. So in this one, we have 1 homozygous dominant, we have 2 heterozygous, and we have 1 homozygous recessive. What does our phenotypes look like? Well, we have 3 green and 1 yellow. Geno versus pheno. So we have 3 green and 1 yellow. This is what Mendel was seeing. He was seeing this 3-to-1 ratio when he was mixing these heterozygotes, these F1. This could be another example of those F1 plants. So, genotypic ratio versus phenotypic ratio, I've already been showing you a little bit just to get you guys comfortable with it. So let's do big B times little b. And what is the genotypic ratio of this cross? Well, a genotypic ratio is going to be 0 homozygous dominants, 4 heterozygotes, and 0 homozygous recessives. For the phenotypes, it is going to be, let's say B is wrinkly, like big B is round and smooth and little b is wrinkly. For the same cross, we're going to have a 4 round to 1 to 0 wrinkly. Excuse me, there's no little b in this. So that's how these ratios will work out. The last thing we're going to talk about is a test cross. This is used to determine genetics of an unknown individual. We'll cross the individual with an individual that has a recessive trait. The nice thing about a recessive organism is that it will always have the little x's, little x, little x. The test cross will either be, and I'm going to use two colors here, it will either be big X, or it will be big X, little x. This is because what we're really testing here is this organism homozygous, dominant, or heterozygous? And from this, we can tell, like if it's a homozygous dominant, all of these will have a big X in them. They will all be that trait. But with the purple one, big X, little x, big X, little x, what ends up happening is half of them will show the dominant trait and half of them will show the recessive trait in a 50-50 split. And that's how we know that we have a heterozygous. If it was 100%, if it was a 4-to-1 instead, we would 4-to-0, excuse me. Instead, we would know that it was big X, big X. With a 50-50, we know it's big X, little x. Now you have an assignment today on this. If you have any questions, circle them. Try to complete it all. And then if I'm back tomorrow, we'll talk about it. Have a wonderful rest of your day.